Swayam Prabha. Digital India. Educated India.
Okay, today's topic is primarily focused on chromatin remodeling. How chromatin remodeling affects gene expression in eukaryotic cells is what is going to be discussed in this particular lecture today. Again, to recapitulate what we have discussed so far, again I will not start all of them. We started from eukaryotic RNA polymerases, then discussed about core promoter elements, then we discussed about various general transcription factors, how they in conjunction RNA polymerase form a pre-insertion complex and results in basal levels of transcription. Then we discussed about enhancers and upstream activation sequences and how transcriptional activators and transcription repressors through their DNA binding domains and either transcription activation domains or transcription repression domains bind to specific sequences in the upstream regions of promoters and bring about either increase in levels of transcription or decrease in levels of transcription. Then we very briefly discussed about the role of chromatin. And discussing the role of chromatin in gene regulation, we focused primarily on how post-translation modifications of histones play a very important role when, for example, histones are phosphorylated or when histones are acetylated, it results in positive regulation of gene expression, and when histones are deacetylated and when histones are methylated, it results in condensation of chromatin and results in repression of transcription. And based on the studies on histone methylation, demethylation and phosphorylation, we discussed what is called as the concept of a histone code, where specific post-translation modifications can be a signature motif for predicting certain gene functions. We can clearly say, for example, there are if a particular lysine residue is methylated or acetylated, you can predict whether that chromatin is going to be euchromatin or heterochromatin. This is what is people are now talking about histone code. It is still not perfected, so many people think that we are still not ready to talk about a histone code. And then we discussed in the last class about another important area of gene regulation, about how DNA methylation plays a very important role in the regulation of gene expression, and how among the 4 bases of DNA, cytosine can actually be methylated by enzymes called DNA methyltransferases, and methylation of this cytosine then is methylated cytosines are recognized by specific methyl CpG binding proteins, and these methyl CpG proteins, when they bind to the cytosine residues of the DNA, can now attract negative regulators of gene expression like the histone deacetylases or histone methyltransferases, and this results in repression of gene expression. And most importantly, we discussed DNA methylation in conjunction with histone modifications constitute what is called as an epigenetic code of gene regulation. Okay, this is what the crux of what we have discussed so far, and today we are going to discuss about another very important and a new area of investigation in the area of eukaryotic gene regulation, that is chromatin structure can also be modified not only by modification of the histones or histone tails, but also by an ATP dependent chromatin remodeling mechanism. This is what is going to be the focus of today's discussion. So far we have studied chromatin remodeling involving post-translation modifications of histones and DNA methylation, and how DNA methylation in conjunction with chromatin modifications of histones can result in regulation of gene expression. This is what discussed. Today we are going to discuss how there is also what is called as an ATP dependent remodeling of chromatin, which is very different from covalent modifications of histone tails. This has nothing to do with covalent modification of histone tails, but there is an ATP dependent chromatin remodeling machinery which actually can also bring about which can alter chromatin structure and therefore bring about chromatin regulation. Let's see what happens now in this cartoon. Let's say we have a double stranded DNA and we have histones which are regularly placed in a normal double stranded DNA. Now there are proteins, there are actually multi-protein complexes which together are now called as chromatin remodeling complexes, and when these chromatin remodeling complexes bind or recruited to the chromatin, and these chromatin recombinant complexes in an ATP dependent manner, when ATP is converted to ADP and PI, energy is released. This energy derived from the ATP hydrolysis is actually used by chromatin remodeling complex to actually displace histones from a DNA, and as a result you generate what is called as a nucleosome free regions, and these nucleosome free regions can now expose binding sites for transcription factors or the pre-initiation complex assembly, resulting in activation of transcription. So, this ATP dependent chromatin remodeling has become another important mechanism by which transcription regulation is brought about in eukaryotes. So, displacement of nucleosomes during chromatin remodeling leading to generation of a nucleosome free region in chromatin, resulting in the exposure of certain DNA binding sites. So, the transcription factors can now come and bind and then initiate gene regulation is another very important area in the regulation of gene expression in eukaryotes. In addition to displacement of nucleosomes, chromatin remodeling utilizing ATP can also involve several other mechanisms. For example, instead of actually displacing nucleotides, there are certain chromatin remodeling proteins which actually slide nucleosomes little bit further. You can see here histone octamers may actually slide along DNA, thereby exposing specific sequences for interacting with other clones. For example, here there is an histone octamer, and by the influence of a chromatin remodeling complex, this nucleosome may actually be pushed further down, and it can even be slided further down. So, by sliding these histones little bit away, it can even be as little as 1 base pair, it can even be slide as little as 5 base pairs. By just moving or sliding histones little bit, you may be exposing a transcription factor binding site. Therefore, these sites which were not accessible to certain transcription factors now can become accessible to transcription factors. Therefore, genes can either be activated or repressed depending upon an activator or repressor has been recruited. So, nucleosome displacement or nucleosome sliding and even adjusting the spacing between histone octamers, all these are major mechanisms by which chromatin remodeling proteins bring about changes in chromatin structure which affect gene regulation. Here is a cartoon, for example. Here is a DNA in which nucleosomes are regularly spaced, and there are chromatin remodeling machinery which actually alter this nucleosome spacing, thereby exposing certain sites which was previously not accessible to transcription factors or transcription machinery. Therefore, proteins which could not bind when the nucleosomes are regularly spaced can now bind after the chromatin remodeling machinery has altered the nucleosome spacing. So, there are a number of mechanisms by which the nucleosome positions can be altered in an ATP dependent manner by certain protein complexes, and these are known as chromatin remodeling machines. So, what we are going to discuss today is that what are these chromatin remodeling machines and how they influence gene regulation. Now, the question we are asking is that why do you need ATP for moving this nucleosome around, either for sliding nucleosomes or for displacing nucleosomes? Why do you require ATP? As you know, bending DNA around nucleosomes depends on the DNA sequence to some extent, and it has been very well shown by a number of experiments that there are certain sequences depending upon the DNA sequence, certain DNA sequence can bend much more than other sequences. In fact, this DNA bending has become a very important aspect of gene regulation, because when you have long stretches of DNA, there have been examples where certain transcription factors can actually bind several thousand kilobases upstream region, but can still affect the rate of initiation at the promoter region. Now, the question is how transcription factors which are binding so far upstream can actually influence the rate of transcription initiation. It actually turns out this DNA which is in between can actually loop out, and as a result this transcription factor which is binding very far can actually brought in a very close proximity to the general transcriptionary. Therefore, it can interact with some of the general transcription factors influencing regulation. So, this DNA bending plays a very important role in the transcription, and many a times the nucleotide sequence of the DNA plays a very important role whether the DNA can be bent or not. Now, nucleosomes we all know have a very strong affinity for DNA, because histones are positively charged, DNA is negatively charged, and therefore histones bind DNA very tightly, and many times some of these histone DNA interactions may also be sequence specific. So, this limited specificity of histone DNA interactions has been utilized by nature to organize chromatin in a specific manner. Now, otherwise you cannot organize nucleus. There is what is called nucleosome facing, there is a 123 base pair DNA which is wrapped around neuron, there is a nucleosome free region, and nucleosomes are regularly organized in a very specific manner inside the DNA. Therefore, there is some kind of an organization of chromatin, and this may be primarily mediated by the histone DNA contacts and histone DNA interactions. And in nature, the nature has divided this mechanism in such a way that a chromatin landscape is energetically conducive. Sometimes it may be conducive for gene expression, sometimes it may not be conducive for gene expression. Okay. Now, if this landscape remains as such, then modulation is not possible. It's like a traffic movement in a traffic junction. You need to have these traffic lights, because depending upon the traffic movement, you have to either give a green light or a red light. So depending upon whether gene has to be expressed in high levels or a gene has to be expressed in low levels, chromatin structure has to be altered. Okay, if the chromatin structure remains as such all the time, then modulation of gene expression is not possible. That's what we have discussed in the last few lectures. So this chromatin landscape, which is determined by specific histone DNA interactions, has to be modulated by a number of mechanisms, including histone post-transfer modifications, which we discussed earlier. So the landscape, the chromatin landscape has to be changed to suit gene expression requirements. That means the histone DNA interactions has to be disrupted, and this requires energy. So if we have to move around or replace histones for different regions, in addition to adding negative charge or positive charge on histones by post-transfer modifications, if we have to remove the histones or move around histones, you need to consume energy to move these nucleosomes around. Now, the first ATP dependent chromatin remodeling complex was discovered in the budding yeast Saccharomyces cerevisiae, and is actually called as SwaySNF complex. What is the SwaySNF complex? Sway actually means it was actually discovered in yeast mating type switching. SWI stands for switching of mating types in yeast. So there is a gene called HO which encodes an endonuclease, which plays a very important role in regulation of mating type switching in yeast. And in fact, for this mating type switching, they found out if you have mutations in the SWY gene, then mating type switching is impaired, clearly indicating that there is some kind of a little link between the SWY proteins and mating type switching in yeast, and that's how this SWY complex was actually broken. Another important component is called SNF. SNF actually stands for sucrose non-fermentable. During sucrose fermentation, people have realized, again, if you have mutations in this kind of a chromatin remodeling complexes, again genes involved in sucrose fermentation, metabolism of sucrose fermentation is affected because of the gene expression changes. Therefore, the first ATP dependent chromatin remodeling complexes were designated as SwaySNF complex, and this SwayFNF complex encodes genes which affect regulation of yeast mating site switching and genes involved in sucrose metabolism. And people have then went ahead and purified all the components of these chromatin remodeling complexes, and as you can see, these chromatin remodeling complex, namely SwaySNF complexes, are a multi-protein complexes, and often the complex is very huge. It can go up to 2 megadaltons, and it contains a number of subunits which are actually designated as SWY1, SWY2, SWY3, SWY5, and so on and so forth. So chromatin remodeling complexes are multi-protein complexes which play a very important role in the regulation of gene expression by modifying chromatin structure. But the number of chromatin remodeling complexes may not be very high. There is only about 100 to 500 molecules of SwaySNF may be present per cell, and as a result, only about 5% of the total genes in the yeast cells are actually expression is regulated by the SwaySNF complexes. So these ATP and chromatin remodeling complexes are involved in, have a limited role in the regulation of gene expression. Not all the genes are regulated by chromatin remodeling complexes, only a subset of genes are actually affected by this chromatin remodeling mechanism. For example, if you talk about the regulation of genes involved in galactose metabolism, we all know, for example, the GAL4 transcription factor plays a very important role in switching on genes involved in the galactose metabolism. And this GAL4, if it has to activate genes involved in galactose metabolism, requires this SwaySNF complex. So there are certain transcription factors where if they have to act or activate gene expression, they primarily do it by recruiting chromatin remodeling complexes. So certain transcription factors bring about gene activation by recruiting histone modifying enzymes, whereas certain transcription factors regulate gene expression by recruiting chromatin remodeling complexes. And there are many instances a combination of both histone modifications as well as chromatin remodeling machinery together bring about regulation of gene expression of a particular promoter. Okay. Now, the SwaySNF binds DNA and nucleosomes in a ATP dependent manner with a very high affinity. The KD of SwaySNF complex for DNA and nucleosomes in the nanomolar range. So they bind very tightly. So about 1000 ATP molecules are shown to be consumed per minute. So it's a very energy demanding reaction. Okay. After binding DNA, the SwaySNF complex starts an ATP dependent remodeling chromatin by actually moving around histones or even displacing histones. The key component of these chromatin remodeling complexes, I told you there are a number of protein complexes in these chromatin remodeling complexes. One of the key components of this chromatin remodeling complex is a protein which contains a ATPase activity. That means it is capable of hydrolyzing ATP to ADP and PI, and since the entire process requires energy, this ATPase activity is very important for the entire remodeling process. So, one of the key components of the chromatin remodeling complex is an ATPase. For example, in the SwaySNF complex, a protein called as SWY2 is actually a DNA stimulated ATPase, which actually plays a very important role in the entire process of chromatin remodeling. Now, chromatin remodeling complexes are classified based on the protein motifs found in addition to the ATPase domain. So, they also contain number of protein motifs and also how the ATPase domain itself is structured. Remember, if once you start isolating or identifying these chromatin remodeling complex number of organisms, you have to organize them in a specific format so that you can understand. You have to group them based on, you need to show whether the human enzyme is related to yeast enzyme or an yeast enzyme is related to Drosophila enzyme and so on and so forth. So, you need to identify certain pattern. Therefore, these chromatin remodeling complexes from different organisms have been grouped depending upon specific structural motifs they have and also depending upon the ATPase domain. If, for example, a Drosophila chromatin remodeling complex ATPase domain is homologous to that of a human, then you put them under the same family, so on and so forth. So, this classification is purely structured, designed to make it easier for us to sort them all out. Otherwise, it becomes very difficult to understand and relate the various studies being carried out in different organisms, and this classification is not necessarily based on any functional criteria. I have just given one example of showing how the various chromatin remodeling complexes have been classified into different families. For example, there is what is called as a, if you take for example the SWY SNF2 ATPase superfamily, remember the SWY SNF chromatin remodeling complex are the first chromatin remodeling complexes which were discovered in Saccharomyces cerevisiae, and this family now consists of a called as a SWY2 SNF2 subfamily. There is something called a ISW1 subfamily, CHD or MI2 subfamily, INO80 subfamily, and so on and so forth. And each subfamily, the homologs have been identified in yeast, human, Drosophila, and so on and so forth. And there is also all kinds of nomenclature. Again, I don't want to confuse you at this stage about this nomenclature. Just to give an example what these things actually mean. For example, when you call a chromatin remodeling complex as NURF, it actually means nucleosome remodeling factor. Nucleosome remodeling factor, NURF. That's what it means. Similarly, there is a nucleosome remodeling complex called CHRAC, crack. It actually means chromatin assembly complex. CHR stands for chromatin assembly complex. So, these are all the abbreviations given to identify a different chromatin remodeling complex in different organisms. Similarly, ACF stands for ATP dependent chromatin assembly and remodeling factor, ACF. Now, in different organisms, different chromatin remodeling complexes play different roles. For example, if you take this NURF remodeling complex, the nucleosome remodeling factor is involved in the transcription activation of certain homeotic genes in the case of Drosophila. It has been very well studied. So, different chromatin remodeling complexes are involved in the regulation of different sets of genes in different organisms. And these names, these abbreviations are actually mean that they have a certain meaning. There are also different requirements for various nucleosome remodeling complexes. For example, if you take the SwaySNF complexes, it has a maximal activity on the DNA alone, without not necessarily chromatin, even DNA alone they have a maximum activity. If you now take for example MI2, it requires a nucleosomal structure for its function and it does not require any histone tails, whereas if you take ISW1, it is partially activated by DNA. In the case of SwaySNF, the DNA itself can activate the ATPase activity, whereas here it requires a nucleosomal structure, whereas here it is partially activated by DNA and the DNA ATPase activity is further stimulated by nucleosomes and it requires histone tails. So, different chromatin remodeling complexes have different requirements for their remodeling activity. I am just giving 3 examples here. Now, how these chromatin remodeling complexes move around histones or nucleosomes also vary from one chromatin remodeling complex to another. For example, if you take the ISW1, it actually moves nucleosome from the central part of the nucleosome to the terminal position. They move the histones from the central part of nucleosome to the periphery. whereas if you take crack, which also contains ISWI, it actually facilitates the movements of histones from terminal to the central region of the histones. So how they actually rearrange histone nucleosomes also depends upon, varies from one chromatin remodeling complex to another. The structure of all these chromatin remodeling complexes and their members have been very well studied. I have just given one example here. For example, if you have there is a chromatin remodeling complex called Brahma. If you look at one of them, if you look at various components of it, it has an ATPase domain, and as I mentioned, the ATPase domain is very important for the remodeling activity, and it also has a bromo domain. And if you go back and recall some of our histone modification studies, the bromo domains are very important for recognizing acetylated histones. So the bromodomain proteins, one of the important activities of HATs is to recruit bromodomain containing proteins. So proteins recognize acetylated histone through their bromodomains. So HATs actually contain bromodomains. So it also contains acetylated with, because this remodeling complex also contains the bromodomain, it can actually interact with the acetylated histones. So this is how the chromatin remodeling and histone acetylation can be brought together for a specific gene expression process. What are the mechanisms by which this ATP remodeling is brought about? There is a very nice article in Journal of Cell Science. These models are a little bit complicated and very difficult to explain without figures, but since there are certain copyright issues in bringing some of these model figures into this lecture series, I have not been able to discuss in detail about the mechanistic insights of many of these models. So I have just very briefly mentioned. For example, there is something called a twist defect diffusion model, where in this model small local alterations from the mean twist defects propagate around nucleosome. That is what this model talks about. Very difficult to explain these models without figures, but I suggest you refer this article in Journal of Cell Science, which very nicely describes all these models of ATP remodeling using very nice cartoons and very nice figures. There is also what is called as cis-trans chromatin remodeling. Now the nucleosomes need not be moved around only within a particular chromatin fiber. There are also what is called a trans remodeling, where histone or a nucleosome from one chromatin or one DNA template can be transferred to another chromosome or another chromatin fiber. So in trans. So a cis remodeling, trans remodeling, all these are possible when you talk about ATP and chromatin remodeling. There are also chromatin remodeling not only just involves moving around histones, it also involves what is called say displacement of histones. There can be exchange of histones. For example, dimers of H2A or H2B can be rather easily exchanged in and out of nucleosomes, and entire histone octamers including H3 can also be displaced or exchanged under certain circumstances. Either specific components of a nucleosomes can be exchanged, or the entire nucleosome octamer can be exchanged, and there are all kinds of variations when you talk about chromatin remodeling in different organisms. And very importantly, chromatin remodeling also involves what is called a variant histone replacement. This also we have discussed in some of the earlier classes, where I have told you that there are variants of histones which actually can be replaced in certain specific states of cell cycle. For example, there are, in addition to histone H2A, H2B, H3 and H4 and H1, there are many variations of these H4 histones occur, and many of these histone variants are expressed, for example, during germ cells, during gametogenesis. Certain histones are replaced by what are called testis specific variants and so on and so forth. So many of these histone variants actually exist, which are very different from the classical core histones, and they are often expressed in a cell type specific manner, and these variants of histones can be incorporated into chromatin in a DNA replication dependent manner. For example, a chromatin remodeling complex known as SWR1 promotes the exchange of H2A from a nucleosome with a histone variant called H2AZ. So it is not just shuffling around histones or moving around nucleosomes. The classical histones can be replaced by very specialized histones by certain chromatin remodeling complexes. And in addition to transcription and regulation of gene expression, the chromatin remodeling also plays a very important role in DNA repair. For example, certain chromatin remodeling complex such as the INO80 is actually involved in DNA repair processes as well. So chromatin remodeling is important not only for regulation of gene expression, but it also has a very profound role in a number of other cellular processes including DNA repair. So the important point that I want to mention here is that just as you have transcription factors which recruit histone modification enzymes like histone acetyltransferases, histone deacetylases, histone kinases and so on and so forth, there are also many transcription factors which are capable of recruiting chromatin remodeling complexes. So, by recruiting these chromatin remodeling complexes, it can bring about regulation of gene expression. So, just as certain transcription factors recruit histone modifying enzymes such as HATs, HDACs, histone methyltransferases or DNA methyltransferases, there are certain gene specific activators like GCN4, GAL-VP16 etc. Instead of recruiting these histone modifying enzymes, they can actually recruit SwaySNF complex directly. So, the chromatin structure can be altered either by post-translational modification of histone tails by recruiting these histone modifying enzymes, or chromatin structure can also be modified by recruiting chromatin remodeling complexes. So, it varies from one particular context to another context. For example, the purified SwaySNF complex actually stimulates the binding of GAL4 acidic activation domain to nucleosomal DNA by 10 to 30 fold. So, the binding of a transcription factor to nucleosomal DNA can be very dramatically enhanced by some of these chromatin modifying complexes because their affinity to some of these transcription factors. So, one of the major mechanisms by which transcription factors regulated gene expression is by direct recruitment of chromatin remodeling complexes. These chromatin remodeling complexes using ATP can now move around or shuffle around or remove nucleosomes, facilitating the binding of transcription factors or assembly of pre-initiation complex, resulting in activation of transcription. So, altering chromatin structure and gene expression by a combination of histone modifications and ATP dependent chromatin remodeling. Remember, although we are studying all these processes in a very sequential manner, initially we talked about histone modification, then we talked about DNA methylation, then we are now talking about chromatin remodeling, but in VIO these things did not happen in a sequential manner. Many of these happen in a very concerted manner, and in any given promoter, for example transcription activation from a promoter may initially involved in acetylation of histone, and this acetylation of histone may actually recognize a HAT complex, and this may also then recognize involve the lead to the recruitment of a chromatin remodeling machinery, and all these things in conjunction can result in the efficient recruitment of a pre-initiation complex, resulting in faster turnover of RNA polymerase, resulting in enhancement of rate of transcription. So, all these processes may actually act in concert to promote transcriptionation from a particular promoter. For example, targeted acetylation of histone H3 by SAGA. As you remember, SAGA is a HAT containing multi coactivator which was originally identified in the yeast. So, it is a HAT containing coactivator. Target acetylation of histone H3 by SAGA stabilizes its binding and that of a targeted SwaySNF chromatin remodeling complexes, and this actually requires the bromodomains of GCN5 and SY2 respectively, which bind to acetylysine. So, some of the members of the SAGA, GCN5 is a member of the SAGA complex, and the GCN5 actually and the SY2 can now recruit a chromatin remodeling complex. Therefore, histone acetylation in combination with chromatin remodeling can actually involve in the regulation of gene expression by the SAGA complex. So, it appears different promoters may use different combinations of histone modifiers and chromatin remodelers. Like I said, if a particular gene has to be repressed, then a histone deacetylase in combination with specific chromatin modifier may bring about tighter assembly of histones, and therefore transcription can be shut off. On the other hand, if a particular gene has to be activated, a histone acetylase or a histone kinase can, in conjunction with a specific chromatin modifier or remodeler, can bring about activation of gene expression by moving around or removing histones from the promoter. The take home message is that there is nothing like a general rule. Now, what kind of a combination of histone modifying enzymes and histone chromatin remodeling factors are working together varies from promoter to promoter. In fact, the reason I am emphasizing this point is that it is, if you now go into the literature or read some of the research articles, you may be flooded with examples and examples and examples, each describing different combinations of histone modifying enzymes, different combination of chromatin remodelers, how they work together and bring about activation or repression of transcription. And it is impossible to discuss all these examples. So, maybe we will discuss some specific examples as we go along in this course, but there are so many variations, so many combinations, it is very difficult to discuss all those things. So, remember, a combination of histone modifying enzymes and chromatin remodelers together may work, and this combination ultimately may decide whether the gene has to be activated or gene has to be repressed. Certain coactivators or coactivator complexes contain components that recognize a specific histone modification as well as those they contain that can remodel chromatin. Okay. For example, the HURF complex, which is a chromatin remodeling complex, contain what is called a BPTF, which can recognize a methylated lysine of histone H3 as well as SNF2 ATPase. So, here is a multi-protein complex, a coactivator, and one component of this coactivator is a histone which can recognize methylated histones. Another member of this coactivator complex is actually a chromatin remodeling complex. So, you can see there are multi-protein complexes which contain both histone modifying activity as well as or a histone it can recognize both a histone modification and also containing a component of a chromatin remodeling complex. So, all these things act in concert ultimately to bring about the regulation of gene expression. The chromatin remodeling may also depend upon the nucleotide sequence of a particular promoter and certain promoters are more susceptible or sensitive to chromatin remodeling than other promoters. The one best example that I can give you is that certain AT rich regions are often nucleosome free and therefore promoters containing such AT regions are better primed for transcription activation. Okay. So, the nucleotide sequence may also, there are certain nucleotide regions or certain promoter regions or certain nucleotide sequences which may be more amenable for chromatin remodeling than other promoter sequences or promoter nucleotide sequences. Transcription factor binding sites are often present in nucleophorosome free regions and therefore does not require chromatin nucleotide displacement. So, there are many instances actually where the transcription factor binding sites actually contain what is called as DNA zone hypersensitive sites. That is, these sites are actually nucleosome free and therefore, when we take nuclei and then treat them with DNAs, certain promoter regions can be very efficiently cleaved by DNase 1 because there are no nucleosomes there and therefore, transcription factor can actually bind them very efficiently. Therefore, these are nucleosome free regions, therefore, no chromatin remodeling is necessary here. Transcription factors can cleanly bind and these promoters can be activated very efficiently without any chromatin remodeling without any need for chromatin remodeling. And it is often found that nucleosome density at promoter regions is typically lower than that in the coding region. Remember, any statement I make like this, there will always be exceptions, but we are talking about very general or at least consensus, at least in majority of the case it has been found. If you look at the nucleosome density in a promoter region versus a coding region, normally the nucleosome density in a promoter region is much less compared to the nucleosome density in a coding region. Now, there are a number of examples in the literature to actually describe how chromatin remodeling can actually bring about regulation of gene expression. I have just given one example here just to tell you how a simple 5 base pair sliding of the histones along the nucleosome can expose a transcription factor binding site resulting in transcription activation. It is a very nice example. You can read up a little bit more because it is a very nice example to very well studied example to understand chromatin remodeling and regulation of gene expression. Now, we all have studied in the previous few lectures about a nuclear receptor called glucocorticoid receptor. We have discussed very well how in response to glucocorticoid hormone, a glucocorticoid receptor which is present in the cytoplasm now undergoes conformational change, dissociates from HSP90 and this glucocorticoid receptor then dimerizes, then goes inside the nucleus, bind to specific sequences, upstream sequences called as glucocorticoid response elements and then this results in activation of transcription. So, all genes which are responsible to this glucocorticoid hormone contain these glucocorticoid response elements and the glucocorticoid receptor now goes and binds there and in the presence of glucocorticoid hormone receptor, glucocorticoid hormone, the transcription activation domain now interacts and recruits a specific coactivators resulting in activation of transcription. Now, very beautiful experiments have been conducted to actually understand how the glucocorticoid receptor functions in a chromatin template. For example, in the case of mouse mammary tumor virus called as MMTV, it has what is called as a long terminal repeat. These retroviruses and these mouse mammary tumor virus contain what is called as a long terminal repeat which actually contain promoter regions for transcription of all the viral genes. Now, this MMTV LTR contain 6 partly palindromic sites and each of them is bound by one dimer. Remember, glucocorticoid receptor recognize 2 half sites AGA, ACA, 3 nucleotide spacing TGT, TCT. This is the binding site for a glucocorticoid receptor. So, if a promoter has AGA, ACA, 3 nucleotides TGT, CCT, remember AGA, ACA, TGT is a palindrome. TGT, CCT if you read in the opposite strand is AGA, ACA. So, you have AGA, ACA and AGA, ACA on the 2 opposite strands and TGT, CCT on the half sides. So, you have 2 half sides AGA, ACA, TGT, CCT separated by 3 nucleotide spacer and this serves as a binding site for one dimer. So, one monomer recognizes AGA-CC on one strand and another monomer recognizes the AGA-TC on the other strand and the receptor binds as a dimer. Okay. So, if there are 6 such sites, 6 palindromic sites, then 2 plus 2, 4 act as a binding site for a 2 glucocorticoid receptor dimer. Now, see, let us read this. So, that is a very interesting experiment. In addition to this binding site for glucocorticoid receptor, the MMTV LTR also has two adjacent sites for the binding of nuclear factor 1 and OCT1. These are also two important transcription factors which are required for transcription activation of the MMTV promoter. Okay. Now, it turns out both the GR and this nuclear factor 1 cannot bind to MMTV LTR simultaneously on a linear DNA template. Now, if you have a linear DNA template and you have these glucocorticoid response elements and you have the binding site for nuclear factor element, but once the glucocorticoid receptor binds, because of steric hindrance, the adjacent site for nuclear factor 1 cannot be occupied by nuclear factor 1. Therefore, transcription activation is not very efficient on a linear template. But and in the absence of hormone, glucocorticoid receptor can only bind 2 of its 4 binding sites. So, in order to bind to the remaining 2 sites, a change in rotational positioning on the nucleosome is required. That means, the nucleosome, the histones has to slide a little bit so that the other 2 sites can also be exposed. So, when hormone bound glucocorticoid receptor binds to MMTV LTR chromatin template instead of a naked DNA template where it binds to a chromatin template, it results in the recruitment of SWY-SNF complex. So, the hormone induces a conformational change in the transcription activation domain of glucocorticoid receptor, resulting in the recruitment of the SWY-SNF complex. This results in chromatin remodeling such a way that the histone H1 is dissociated, and as a result the binding sites for NF1 and OCT1 is now exposed on the other side of the chromatin, and this results in the binding of NF1 and OCT1 can now bind along with glucocorticoid receptor, and this results in the recruitment of TF2D and ultimately transcription activation. So, you can see you have glucocorticoid receptor and you have NF1, they cannot bind to their binding sites on a naked DNA template because of the steric hindrance, but they can bind very efficiently when the same DNA is organized in the form of chromatin, and by recruitment of a chromatin remodeling complex and displacement of histone, you can now access, both these factors can access the binding site, resulting in very efficient transcriptional activation. In fact, there are very nice experiments that have been done to actually show NF1 binding to a chromatin template can be footprinted only when you add hormone. Whereas the absence of hormone, NF1 cannot be bound, but the moment you add hormone, the hormone bound glucocorticoid receptor now recruits a chromatin remodeling complex and therefore facilitates the binding of NF1. So, just to tell you that how moving nucleosomes around can actually expose certain transcription factor binding sites and therefore facilitate synergistic action of transcription factors, which in concert can result in activation of transcription from chromatin templates. And this is another example here. Interactions between transcription factors and remodeling complexes provides very important sign to the mechanism of actions. This is one mechanism what I told you so far, how the glucocorticoid receptor binding and recruitment of chromatin remodeling complexes now facilitates the removal of histones and exposes signing for an adjacent transcription factor to bind, and together it results in the recruitment of TF2D and transcription activation. And in the case of yeast cells, for example, there is a transcription factor called SWY5P, which is not a chromatin remodeling complex, which is a regular transcription factor, and this activator is actually required for transcription activator of the HO locus. Remember, HO is a gene that encodes endonuclease, which is required for mating type switching in yeast cells. Okay. In this particular case, when this transcription factor enters the nucleus at the end of mitosis and binds to the promoter of the HO gene, it results in the recruitment of the SWY-SNF complex. Now, very interestingly, the job of SWY5P seems to be just recruitment of a chromatin remodeling complex to the promoter. Once the chromatin SWY-SNF complex is recruited to the promoter, the SWY5P is released, leaving this SWY-SNF at the promoter. So, the function of this transcription factor in this case is just to recruit a chromatin remodeling complex, that is all. So, you can see there are diverse examples to actually show there are transcription factors like glucocorticoid receptor which recruit a chromatin remodeling complex, but they continue to bind there, and then that facilitates the binding of another transcription factor leading to recruitment of TF2D and activation of transcription. In this case, the only function of the transcription factor like SWY5P seems to be once it bounds, it recruits the chromatin remodeling complex, and then the SWY5P associates, and the chromatin complex take over, brings about changes in chromatin resulting in activation of transcription. And one can ask the question, what is the relevance of studying all this chromatin remodeling? Does it really have any bearing on the human health? Just as I told you that mutations in DNA methyl transferases can result in the manifestation of genetic disorders, clearly indicating that DNA methyl transferases play a very important role in number of genetic disorders and so on and so forth. Similarly, There are also several genetic diseases which have been identified, which have been associated with chromatin remodeling. There is a very nice review in Current Opinion Genetics and Development, which talks about the various genetic diseases which are associated when you have mutations in gene encoding chromatin remodeling complexes, very clearly indicating that chromatin remodeling affects a number of important processes, and when you have mutations in proteins encoding mutations of genes encoding the chromatin remodeling proteins, it can result in genetic diseases, saying that it has a very important physiological function. There are also a number of papers I have listed here which discuss in great detail the role of chromatin remodeling and their effect on gene regulation. So, I am not going to the details of this. So, what I would like to summarize here, so far what I have told you is that chromatin remodeling complexes are nothing but they are ATPases that form a large multi-subunit complex. These complexes actually use ATP, the energy derived from ATP hydrolysis, to remodel nucleosomal DNA, and these are associated with the activation of chromatin, but in some cases chromatin remodeling complexes are also involved in silencing of gene expression. So, in addition to altering chromatin structure involving post-translational modification of histones, we have now brought in a new player into the regulation of gene expression, namely chromatin remodeling. Whereas his post-translational modifications of histones does not require any ATP hydrolysis, whereas these chromatin remodeling machines, which are actually huge multi-protein complexes, they actually use the energy derived from ATP hydrolysis to alter nucleosome positioning. The nucleosomes can slide, or nucleosomes can actually be displaced, or certain classical histones can be replaced with certain variants of histones, and all these possible combinations can result in creating what is called a nucleosome free region in chromatin region, and as a result transcription factors can come and bind to these chromatin templates, resulting in activation of transcription or repression of transcription. The other important point I have mentioned here is that, just as I mentioned in the previous class, that histone acetylation, DNA methylation and many other modifications they act in concert. There are also examples wherein chromatin remodeling complexes in conjunct with histone modifiers act in conjunction to either activate or repress the transcription from a particular promoter. So, we should never get the impression that just because we are studying each one of these topics in a different classes does not mean that these all act in isolation. In vivo, all these processes are combined together and they all occur together and they complement each other, and one process is dependent on another. For example, in many cases the acetylation of histones can actually lead to the recruitment of a chromatin remodeling machinery along with the histone acetyl transferases. In many cases, the coactivator complexes may contain both an histone acetyl transferase as well as a chromatin remodeling ATPase together, indicating that all these things actually act together and one process leads to another, and ultimately together a combination of these events is what ultimately determines the activation or repression of transcription of specific genes. This is what we have discussed now. There are also a number of experimental techniques that people use for studying this kind of a chromatin structures and also identifying the role of these chromatin modifying enzymes. Although I will just mention one important technique which people in the field normally use. If you want to really identify the organization of nucleosomes, whether the DNA is really organized in the form of nucleosomes, you do what is called as a very important experimental technique, which is called as the macrococcal nuclease digestion. Okay, I have just taken this cartoon from this particular paper where they actually demonstrated how you can distinguish a euchromatin region in a nucleus from a heterochromatin region. Suppose I want to find out, I am interested in a specific promoter or a specific gene, and I want to find out whether this promoter region exists in the euchromatin region or heterochromatin region of the in the nucleus. What I do is that I take the nuclei and subject this nuclei to what is called as a digestion with macrococcal nuclease. Now, as you know, the nucleosomes are very spinely, very evenly, very nicely organized, and when you now take such DNA and then treat that macrococcal nucleus cleaves only the interhistone region, between the histone octamers at low concentrations, and as a result it leaves only these interhistone regions or internucleosome regions. Therefore, you create nicks only at these regions, and therefore you get what is called a very nice ladder. So, what you do is that you do the macrococcal nucleus digestion and then isolate DNA and then do a southern blotting by making a radiolabel probe of your promoter region of your interest and probe this blot with this particular probe, and the same probe is the same here. But there are two situations, in one case where the gene is getting expressed, another case where the gene is not getting expressed, and very interestingly you see here in this example, when your gene is getting expressed, your promoter is actually present in the form of a euchromatin region, and you can see you no longer see this kind of a ladder pattern here, seeing that nucleosome structure here is altered in the euchromatin region, whereas the gene is not expressed, you see a very nice nucleosome pattern here, because the MNase cleaves only in the intra-optimum region, you get a very nice ladder pattern. So, using techniques such as these things, you can actually identify whether your gene of interest is present in the euchromatin region or the gene is present in the heterochromatin region. So, what are the implications of all that I have studied, all we have studied, discussed so far is that whether a gene is to be expressed or it should not be expressed depends upon whether the gene is organized in the form of a euchromatin or the gene is organized in the form of a heterochromatin inside the nucleus. And whether the chromatin is in the heterochromatin state or euchromatin state depends on a number of factors. It depends on the methylation of cytosines of the DNA, it depends on the covalent modifications of the histone, it depends on the activity of the chromatin remodeling complexes. So, all these factors collectively contribute to whether your gene has to be transcriptionally active and your gene has to be transcriptionally inactive. And more importantly, there are also now people who have been studying this kind of a chromatin organism inside the nucleus. Many of you might have only talked when you talk about nucleus structure or organization of nucleus, you might have simply studied only about nucleus and nucleolus. Other than these two subnuclear structures, you probably have not studied anything. But recent studies on understanding the organization of chromatin and organization of genes in the euchromatin and heterochromatin have now identified there are a number of subnuclear structures and there are a number of complexes which play a very important role in the organization of genes, and this nuclear architecture plays a very important role in the regulation of gene expression. There are, for example, what are called as speckles, paraspeckles, Kajal bodies. I do not want to explain all these things, we will discuss all these in a later stage, but what I am trying to mention here is that in addition to nucleolus, which is one of the major components inside the nucleus, which is what most of you might have studied, there are also a number of subnuclear structures that are being discovered now, and all these things seem to be playing a very important role. There are many instances where immediately after transcription the RNA is not exported out. The RNA is actually stored in specific subnuclear structures for some time and then it is taken out. Okay. So, in addition to transcription per se, there are also a number of post transcriptional processes which regulate whether the RNA which has been transcribed has to be translated and a protein has to be made or not. In addition, the other important point I would like to discuss in the next class is that so far we have been discussing only transcription initiation. Now, what we will discuss in the next class is that once the RNA polymerase kick starts, once the RNA polymerase carbox terminal domain is phosphorylated and the RNA polymerase moves, then there is what is called the transcription elongation. That means the RNA polymerase has to go through and transcribe the entire coding region and the non-coding regions as well, and then in the case of eukaryotes the introns have to be spliced out, the exons have to be joined, and then at the end there is what is called the transcription termination, polyadenylation, and poly A tail has to be added. And what I would like to discuss in the next class is that this transcription initiation, which is primarily carried out by RNA polymerase and the general transcription factors play a very important role not only in transcription initiation, but they also play a very important role in transcription elongation, polyadenylation, mRNA capping, and so on and so forth. Remember, all the eukaryotic messages undergo what is called a 5' capping. There is a 5' cap. The cytosine is modified, the guanine residue of the 5' is modified and is actually called the 5' cap. So, the enzymes involved in 5' capping, the enzymes involved in RNA splicing, the enzymes involved in polyadenylation of RNA, they all interact with the mechanism, the machinery of involved in transcription initiation. So, I do not want you to be under the impression that the function of general transcription factors and RNA polymerase is not just to initiate transcription, but they also have to interact with components of post translational modifications. The machinery involved in transcription initiation, they also interact with capping enzymes, they also interact with splicing enzymes, they also interact with polyadenylation machinery, and all these things are very highly integrated. Ultimately, if a polyadenylated messenger RNA has to come out of the nucleus, all these machinery have to function together and is all integrated. So, what we will study in the next class, after understanding how transcription initiation takes place, how enhancers act, how transcription factors bind and activate transcription, how chromatin plays a very important role in the regulation of gene expression, and how chromatin modifying enzymes, histone modifying enzymes, DNA methylation, all these things in combination contribute to the regulation of gene expression in eukaryotes. In the next class we are going to discuss about how transcription initiation is coupled to transcription elongation, messenger RNA capping, polyethylation, and how all these events are actually integrated with each other. So, let us discuss in the next class about the interlinking or interrelationship between the initiation of transcription, elongation, mRNA capping, and polyethylation, and how all these processes are integrated together. This is going to be the focus of the next class. So, I think with this about 10 lectures, I have now completed one part of this lecture series. This actually forms one major capsule of this entire lecture series on eukaryotic gene regulation, and at the end of these 10 lectures you probably now have a very comprehensive idea about regulation of gene expression, how transcription initiation is affected by transcription factors, and what is the role of chromatin in the regulation of gene expression. So, once we discuss the next class about the integration of transcription initiation between with RNA capping, polyethylation and splicing machinery, we will then move on to understand the mechanism by which RNA polymerase 1 and RNA polymerase 3 initiate transcription. So far in all our discussion we have been focused only on RNA polymerase 2 and protein coding genes. Now, we will then spend 2 classes to understand the mechanism by which RNA polymerase 1 transcription takes place, is transcribes the genes, and RNA polymerase 3, what are the various factors that are associated, what are the important facets of RNA polymerase 1 transcription or RNA polymerase 3 transcription. Then we will move to the next phase of our lecture series, understanding signal transduction processes. We will take examples of each and every one of the transcription factors and study in detail how a signal emanating from the cell surface gets in all the way and then ultimately results in activation or repression of transcription. I think I will end here.